Welcome to AP Biology. This video is on transcription. Transcription is a process that takes place inside the nucleus of the cell. During transcription, a gene is going to be read, and using that gene, a sequence of messenger RNA is going to be created. That messenger RNA can then leave the nucleus, travel to the ribosome, where it will be translated into a sequence of amino acids. Transcription is going to take place in three stages: initiation, elongation, and termination. We begin with initiation. Right before the actual gene, here's our gene shown in dark blue, is a region of non-coding DNA called the promoter. Non-coding means that it's not used to actually build the protein. The first four bases of the promoter region of DNA are TA, and we call this the Tata box. The promoter region of DNA is an example of a control element. Control elements are regions of DNA that are non-coding and are used to control the production of proteins within the body. So they regulate protein synthesis. How does the cell know to transcribe a particular gene? Well, it begins with proteins called transcription factors binding to the Tata box. Here you see a picture of a transcription factor binding to the Tata box. We know that a very common transcription factor in the body are steroid hormones. We know that steroid hormones control protein synthesis within the body. They initiate the process of protein synthesis. And here you see how that's taking place. When a steroid hormone like testosterone enters the body, or enters a cell I should say, testosterone is going to bind to a receptor within the cell, and that testosterone receptor complex enters the nucleus and works as a transcription factor. The transcription factor is going to bind to the Tata box, and this is what initiates the process of transcription. Once the transcription factor has bound to the Tata box, then RNA polymerase can come and bind to the promoter region of DNA. RNA polymerase is going to be the enzyme that actually transcribes the DNA into our sequence of messenger RNA. This first step, in which the transcription factor binds to the Tata box and then RNA polymerase binds to the promoter, is initiation. During elongation, the RNA polymerase is going to read the template strand of DNA, not the coding strand, the template strand. As it travels through the template strand, it's going to lay down all the complementary bases, making its sequence of messenger RNA. Remember that messenger RNA has U instead of T. It does this until it transcribes a sequence of messenger RNA that goes AUAAA. We call this sequence the terminator sequence. When this sequence has been transcribed, the RNA polymerase is released from the DNA and transcription is completed. So if you're looking at this picture on the bottom, here you see our sequence of messenger RNA, and you can see that the protein coding segment extends from here to here. We know that the protein coding segment goes from the start codon to the stop codon, and our terminator sequence actually occurs way after that stop codon. Before the messenger RNA can leave the nucleus, it needs to go through a process called processing. A number of things will take place to the messenger RNA. Number one, the ends are modified. On the 5' side, or the front of the messenger RNA, a 5' cap is added. That 5' cap is made up of GTP. On the back of the messenger RNA, a poly A tail is added. It's a long sequence of adenine. And that's going to kind of work like a telomere. It's going to prevent that messenger RNA from being broken down. Before the messenger RNA can leave the nucleus, it also needs to go through a process called RNA splicing. If you look inside the messenger RNA, there are actual coding regions and non-coding regions of the messenger RNA. The coding regions are called exons. The non-coding regions are called introns. The introns are going to be cut out, and the exons are going to be spliced together before the messenger RNA can leave the nucleus. Messenger RNA can go through a process called alternative RNA splicing. What does that mean? That means that the RNA can be spliced in different locations. It has the ability to change where the exons are and the introns are. And that is going to allow one gene or one messenger RNA to actually code for more than one or multiple proteins. Here you see how messenger RNA splicing actually works. There are small proteins called SNRNPs. It stands for small nuclear ribonuclear proteins. These proteins come together and form a structure called the spliceosome. And you can see what the spliceosome is doing right here. The spliceosome is going to cut at the ends of the intron, and then it's going to splice the exons together. Human beings are the only organism that contain RNA that could actually splice itself. RNA that is splicing itself is acting as something that we call a ribozyme, and this is unique only to human beings. So if you look at a gene, the gene itself has coding regions and non-coding regions. We call the coding regions of the gene the exons, the non-coding regions the introns. The gene itself, the sequence of DNA, goes through transcription and makes the messenger RNA. Then at the level of the messenger RNA, the introns are removed, the exons are spliced together. That messenger RNA then travels to the ribosome where it's translated into a polypeptide. Now if you're looking at that polypeptide, there are different regions, there are different domains. Here you see this polypeptide has 3 different domains, and each domain of the polypeptide is determined by the different exons. This concludes our video on transcription.